Once we are done with theoretical and practical concepts of phylogenetical analysis, we need to know what are different available softwares which can be attained from different places. There are some common characteristics, such as those different softwares, they should be free of cost, they should be available from internet, and then they have some unique or common characteristics, number one common characteristics, and then some unique characteristics which help them to differentiate from each other. So what are those different software or tools for phylogenetic analysis? Again, what was our purpose for phylogenetic analysis was to differentiate species or organisms with their macromolecular information, which was DNA, RNA and protein. So this differentiation can be very helpful if we have some tools which present that information visually or graphically. So there is a broad range of those tools available. Let's see those tools one by one. So again, what will happen, or what are the common characteristics? Number one, they are freeware. Freeware means that they are freely available and then can be downloaded from internet. Then they address unique or certain questions, and they are very good at that. Then several small programs for the whole analysis. Then there is a chance for bug fixing. Bug fixing means if there are some errors, then that can be fixed and returned back. And then there are restrictions. That restrictions mean that it should be specific. It is not a generic program which can do anything, everything. And finally, what we can do, we have some mechanism for input and output of our results. For example, a common input mechanism can be, we can use common softwares which are already present in our system, such as Notepad or TextPad, where we can write our inputs. So now I will give a list of some useful softwares which are there for phylogenetic analysis. यहाँ पर मैं एक चीज़ क्लियर ये कहूँगा कि दिस इज़ नॉट अ वेरी एग्जॉस्टिव लिस्ट, इट इज़ जस्ट अ लिस्ट which consist of commonly used softwares which are there for construction of phylogenetic analysis, and these phylogenetic analysis or these tools, they are following different stages of phylogenetic analysis. For example, number one tool is called APE, which is based on R programming language, which is very similar to MATLAB. So what does it do? It gives us information about phylogenetics and plus evolution against among different species. And what will happen? It has a high list of phylogenetic functions such as multiple sequence alignment, tree construction, distance based, and so on. Similarly, we have Ballet phylogenetics, which is based on simultaneous Bayesian inference of different sequences using their alignment and phylogenetic information. And again, what it does, it converts our input information in trees and then give us feedback. Moving on, we have base traits, which gives information, phylogenetic information against different traits of organisms, and again it is using Bayesian information as inferencing. Then we have beast. Beast is basically Bayesian evolutionary analysis and sampling tree. So what will happen? It will just not only give evolution information, it will also generate some sampling data. So it will say, okay, this information is from this thing and sample was this particular part. Again, another extension is that it will give us a relaxed molecular clock, that it will give us chance that this speciation or radiation did not occur in a strict molecular clock time domain. Similarly, we have bionumerics. Bionumerics, basically it is a universal platform for management, analysis and retrieval of biological data, and it is a very heterogeneous or big platform for different types of data. And with respect to phylogenetics, it roughly consists of all the tree construction algorithms. A list is provided in this slide also. So again, we have cluster W, as we discussed earlier. It is based on progressive MSA algorithm. It has different versions, such as we at the moment we are having cluster omega. Then mega software, which is molecular evolution analysis and genetics information, which is based on again distance measures such as UPGMA, parsimony and maximum likelihood. Then we have tree specific softwares which are for tree construction, such as TreeGen. It is for constructing those trees which are for genes, based on genes or DNA sequences. Then TreeAlign. TreeAlign, basically it is a hybrid software between maximum parsimony and distance based matrices. And finally we have TreePuzzle. Tree puzzle is basically a model based approach which is based on maximum likelihood software.